project pertaining to aerobiology, and owing to research, he has widely traveled in India and abroad. We welcome you, sir, and are honored to have your virtual presence amongst us today. With the permission of sir, now
Okay, thank
Varsha,
you so much,
start
madam.
your
Uh,
presentation.
yes, you can start now.
Yeah. Thank you, sir.
I have done my research work under the guidance of Dr. Ranu Singh from Mata Gujri College and
वर्षा थोड़ा साउंड साउंड बढ़ाओ, आवाज कम आ रही है.
I have done my research work under the guidance of Dr. Ranu Singh from Mata Gujri College and Dr. Akhilesh Ayati from.
नहीं, तुम्हारी आवाज कम आ रही भाई. थोड़ा तुम या तो पास से बोलना पड़ेगा या ईयरफोन लगाओ तुम. हाँ, वो आवाज बहुत दूर आ रही है. या तो आप पास आ जाइए या तो फिर ईयरफोन लगा के उसमें से आप माइक लगा के बात करिए.
हाँ सर. Myself, Varsha Avasti. I have done my research work under the guidance of Dr. Ranu Singh from Mata Gujri College and Dr. Akhilesh Ayati from Home Science College. My topic is phytochemical study on Anora reticulata with reference to its medicinal uses. Now, I am welcome to you, sir, and thank you so much for joining.
लेकिन तुम्हारी आवाज क्लियर नहीं आ रही भाई.
जी सर.
या तो फिर मुझको ईयरफोन लगाना पड़ेगा.
ईयरफोन है तो किसी के पास ईयरफोन इसमें कैसे होगा? यार बेस आ जाएगी आवाज. प्रेजेंटेशन आती है यहाँ से नहीं करना बेटा कुछ. नहीं प्रेजेंटेशन ये कनेक्ट कर दो. अच्छा ये कनेक्ट कर दो. हाँ हाँ ये कनेक्ट कर दो पहले आवाज के लिए. जल्दी जल्दी करो, टाइम नहीं है. तो ऐसे नहीं. हाँ बताओ क्या करना है. बस ये यहाँ पे कर देना. क्या जी मैम. डेस्कटॉप में है ये है बेटा. ये हाँ आ गया मैम. मैम आ गया. First, I give introduction. Phytochemical study on Annona reticulata with reference to its medicinal uses. Rani Durgavati Vishwavidyalaya, Jabalpur, MP. And I am myself Varsha Avasti. I have done my research work under the guidance of Dr. Ranu Singh, Head Department of Botany from Mata Gujri College, and Dr. Akhilesh Ayachi, Principal of Home Science College, Principal of Home Science College. Now I can start content, introduction, material and method. Then third is result, discussion and conclusion. So first is introduction. The medicinal plants are rich in secondary metabolites and essential oils of therapeutic importance. The main advantages claimed for therapeutic uses of medicinal plants in diverse ailments are their safety, in addition being economical, efficient, and their effortless availability. The ethnobotany of the medicinal plants of Jabalpur region is important in understanding the cultures and traditions of Jabalpur people. This study documents one medicinal plant, Annona reticulata. Taxonomy of Annona reticulata, botanical name Annona reticulata, and its common name custard apple and bullocks heart, and its local name is ramphal. Then classification, kingdom Plantae, order Magnoliales, family Annonaceae, genus Annona, species Annona reticulata. The name of the genus Annona is derived from the Latin word annone, meaning yearly produce, referring to the production of fruit. Which part is used in study? I have used seed, stem, fruit, and leaf part of the medicine of this medicinal plants. And this medicinal value is fruit is also used for treatment of diarrhea and dysentery. Leaves used for stomach of children suffering from indigestion. Crushed leaves or paste of the fruit used in ulcers. Then some special features of the plant. Annona reticulata is a small deciduous or semi-evergreen tree. It is best known for its fruit called custard apple. Custard apple rich in vitamin C and riboflavin. Annona reticulata is widely distributed in tropical and subtropical regions. It is belong to Annonaceae family. Annonaceae is a large family under the division of Magnoliophyta, which is comprising about 130 genera and 2,400 species. Then objectives are the present study shall help to broaden the spectrum of the knowledge of indigenous drug used by the tribal people in the treatment of liver disease. Then second is to evaluate botanical and physicochemical parameters of fruit drug as per WHO guidelines, World Health Organization. Evaluation and identification of the primary and secondary metabolites in plant. Then four is to identify the presence of functional groups in plant extract. Screening of different phytochemical extracted from Annona reticulata. Then I will give material and method. So first is collection of plant material. The plant material collected from Jawaharlal Nehru Krishi Vishwavidyalaya in Jabalpur. Then identification of plant were collected from State Forest Institute Jabalpur. Process of extraction. Then first step is washing. Different plant parts were washed with distilled water 2 to 3 times. Then drying. Different plant parts were dried in room temperature. And third step is grinding. The different air-dried parts of Annona reticulata were pulverized to a coarse powder in a mechanical grinder passed from 100 mm massive. Then powder form is kept is in airtight container, and this is the pictures of plant parts of Annona reticulata. So first part is stem, then second part is fruit and seed and leaves. Preparation of extract of Annona reticulata plant part. The different parts of Annona reticulata plant like leaf, stem, fruit, seed, peel of different quantity is added to four solvents, namely distilled water, methanol, ethyl acetate, and petroleum ether. Then Then phytochemical screen, and there is a table and sample, seed, stem, leaf, root, and weight is 10 gram. Each weight is 10 gram. Then phytochemical screening, it refers to the extraction, screening, and identification of the medicinally active substance found in plants. Some of the bioactive substances that can be derived from plants are flavonoid, alkaloid, carotenoid, tannin, and phenolic compounds. It is two types. First is primary metabolites. So, primary metabolites is directly involved in growth and metabolism of plant. And second is secondary metabolites, so organic compound and not involved in metabolic activity of plant, considered as end product of the primary metabolites. Then qualitative phytochemical screening. So first is primary metabolites. Primary metabolites is of three types: carbohydrate, protein, and lipid. And there is some test for carbohydrate. First is Mollis test, Benedict test, and Fehling test. And for protein, xanthoprotic test, pyruvate test, and lipid solubility test, glycerol test, and pseudometry test. Then secondary metabolite, alkaloid. For alkaloid, Mayer test, Wegener test, and Dragon-Troff test. Then saponin, flavonoid resin, and there are three tests for tannin: gelatin test, lead acetate test, and ferric chloride. Secondary metabolites, sterols, for sterols, first is Salkovsky test and Lieberman's test, and cardiac glycoside, killer Kilani test, then triterpenes, coumarin, and anthraquinone. Quantitative phytochemical screening. Phenol test. Take 1.5 ml phonyl cyocalcium reagent plus 1.2 ml of 75% sodium carbonate in a test tube. Then it add zero point 0.3 ml of different plant extract, vortex for 15 seconds, and keep it 30 minutes at room temperature. Then absorbance was measured at 765 nm nanometer spectrophotometer. Then alkaloid test, 5 gram of plant sample placed in 250 ml beaker. Then it add 10% diethyl ether, stand for 4 hours, and filtered. Filtrate concentrated on water bath. Then add NH4 OH of its quarter volume, and mixture allowed to settle. Precipitate collected on filter paper and wash with dilute NH4OH. Then flavonoids, 10 gram of plant sample extracted with 100 ml, then settle in room temperature. Then filtered to mixture, filtrate transferred into water bath, and evaporate to dryness. Then weight of dried filtrate, take the weight of dried filtrate. This weight show percentage of flavonoid. Then test of saponins, take 20 gram of plant sample in a conical flask, then add 100 ml of 20% ethanol. Mixture is heated in hot water bath for 4 hours. Then mixture is filtered, residue extracted with 100 ml of 20% ethanol. then take 20 ml of diethyl ether was added and shaken, and it's shaken. Show a aqueous layer. Then it's add 60 ml and butanol plus 5% sodium chloride. This solution evaporated while water bath. Weight of dried residue shows saponins. Total terpenoids: 10 gram of plant powder soaked in alcohol for 24 hours, then filtered. The filtrate extracted in petroleum ether. Ether extract treated as total terpenoids. Then proximate analysis. I have done this process for knowing nutritive value of plant. For proximate analysis, the fine powder of each dried plant part as seed, stem, leaf, and fruit were used. Then first is a moisture content. Moisture content represent the moisture held inside the cells after complete drying of the plant material. There is a formula for estimation of moisture content, moisture percentage difference in weight upon weight of sample into 100. Then ash content, the percentage of ash represent the inorganic part of the plant cell. It also includes those ingredients which cannot be digested by humans, and formula is ash percentage weight of ash upon weight of sample into 100. Then fat content, estimation of ash contains fat percentage W2 minus W1 upon W. There is W means weight of the sample, and W1 means final weight of the sample, and W2 initial weight of the sample. Crude fiber contain, there is a formula for crude fiber contain W3 into 100 upon W. W is equal to sample weight and crucible weight before ashing. This is W1, and for W2 crucible weight after ashing. Nitrogen contains nitrogen percentage V minus B into N into 14.01 into 100 upon C into D. So there is V is equal to volume of 0.1 and sulfuric acid used for titration for sample, and B is equal to volume of 0.1 and concentrated sulfuric acid used for titration for blank. N is equal to normality of sulfuric acid used, and C is the volume of ammonium nitrate solution, and D is concentration of ammonium nitrate solution. Then protein content. Protein is organic compound made up of amino acid and arranged in a linear chain. Protein is equal to nitrogen in percentage into 6.25. Carbohydrate contain absorbance corresponding to 0.1 ml of text is equal to x mg of glucose. 100 ml of sample solution is equal to into upon 0.1 into 100 mg of glucose is equal to percentage of total carbohydrate present. Then there is a formula for take nutritive value, then protein, fat and carbohydrate. So there is a formula for get nutritive value of these all contain protein, ash, carbohydrate, fat. Then 4 into protein percent plus 9 into fat percent plus 4 into carbohydrate percentage. Then I have done antimicrobial activity. So there are some glassware I have used. Glassware are borosilicate material due to its less thermal expansion are listed below. Petri plates, beakers, conical flasks, test tube, and cartony bottles. Then sterilization of glassware. For sterilization of glassware are washed with water and then wrapped in newspaper. Then autoclaved at 121 degrees Celsius for 15 to 20 minutes. Hot air oven is also used for sterilization at 140 degrees Celsius. Then antibacterial activity. The Muller-Hinton agar media is used for the assay of antibacterial test. The NAM nutrient agar media is used to prepare the slants of isolated culture and kept under refrigeration until use. The plate is prepared of Muller-Hinton agar media and solidified. Inoculate the sample from the culture slant. The cork borer of 7 mm in diameter was sterilized by flaming and used to create 4 wells on each petri dish. Then a small amount of plain agar was poured into wells to seal the bottom of well. Then stock solution of leaf, stem, fruit and seed portion measuring 0.2 ml poured into wells. The plates are allowed to stand for 15 minutes and incubated at 37 degrees Celsius for 24 hours. After incubation, the zone of inhibition is formed and measured in using meter rule in millimeter. Isolation. The collected sample was incubated on nutrient agar plates and incubated on incubator at 37 degrees Celsius for 24 hours by streaking plate method. After incubation, colonies develop on agar plates. A single colony is picked and again inoculated on nutrient agar plates separately and nutrient agar plates separately and incubated at 37 degrees Celsius for 24 hours. Then microscopic examination, the isolated bacterial cultures is examined under microscope for identification of bacteria morphologically. Then antifungal activity, the antifungal screening was carried by well diffusion method and it is similar to antibacterial activity. The different extracts from different plant parts of Annona reticulata were screened for antifungal activity against Candida albicans. ATCC means ATCC procured from American type culture collection. In USA, miconazole 30 μg acts as control antifungal agent. Then this is test process test organism growth on CLED brought cysteine lactose electrolyte dextrose inoculated MHA plate means M for Muller Hinton agar media extracted placed on well on MHA agar zone of inhibition size was measured. Then FTIR spectra for functional group. Fourier transform infrared spectra of pulled fraction was analyzed at the sophisticated analytical instrument facility, Indian Institute of Technology, Bombay, IIT, Powai. So there is a graph, then the transmission and wavelength in centimeter -1 centimeter -1. There is a some peaks for functional group, some shorter peaks and some longer peaks. So primary metabolic screening in primary metabolite screening in seeds of Annona reticulata. So there is a three types of primary metabolites: carbohydrate, protein, and lipid. So molluscus for carbohydrate, molluscus, benedict, and Fehling. Then aqueous carbohydrate is absent in aqueous solution but present in methanol solution and ethyl acetate solution. And protein is absent in aqueous solution but present in methanol solution for xanthoprotic and biuret test and absent in ethyl acetate and petroleum ether. Then lipid, there are three tests for lipid: solubility, glycerol, and Sudon 3. So lipid are absent in aqueous and methanol both, but present in a glycerol test and Sudon 3 test and 3 test of petroleum ether. Then there is a pictures there is a pictures for primary metabolites: carbohydrates, methanol oxide. There is pink, violet pink color present in present in junction of two layers. So it is indicate the presence of oligosaccharide. And then there is presence of some green color. This is indicate the presence of polysaccharides, and Fehling test indicate the presence of reducing sugar. Then this is the pictures of protein test, xanthoprotic test, and biuret test. So protein is present in methanol extracts of seed and for biuret test. So both is methanol extract of seed. Then lipid test. So in methanol extract, white precipitate show indicate the presence of lipid test. And glycerol test, violet pink color is green, some green color is present is for glycerol test of lipid. Then Shudon 3 test, red color indicate the presence of lipid. Then secondary metabolite screening in seeds of Annona reticulata. Alkaloid test, there are three tests for alkaloid: Mayer test, Dragon drop test, and Wegener test. So alkaloid is absent aqueous ethyl acetate and petroleum ether, but present in methanol extract. For Mayer test, Dragon drop, and Wegener test. Saponin test, foam, so saponin is present only in aqueous extract and absent in methanol, ethyl acetate, and petroleum ether. There is flavonoid test. Flavonoid is present in aqueous extract and methanol extract. Then resin test. Resin are present in methanol and aqueous extract but absent in ethyl acetate and petroleum ether. Tannin test. There are three tests for tannin: gelatin test, lead acetate, and ferric chloride test. So tannin is present in lead acetate and absent in ethyl acetate and petroleum ether acetate, but present in methanol extract. Then ferric chloride test. So ferric chloride is absent all four extract of this. And sterol test, there are two tests for sterol: Salkowski test and cardiac glucoside test. So in aqueous solution, Salkowski test is absent, and methanol only in methanol is present. Then cardiac glucoside test. So there is only one test for cardiac glucoside, Killer-Kilani test, and cardiac glucoside is absent in aqueous methanol and ethyl acetate test, but present in only petroleum ether test. Then triterpenes. Triterpenes is absent all four extract: aqueous methanol and ethyl acetate and petroleum ether. Then last is anthraquinone test. So anthraquinone is absent all four part: aqueous methanol, ethyl acetate, and petroleum ether. So there is some pictures of test. Mayer test, there is white precipitate seed methanol extract of seed is present, and Dragon drop test for reddish brown color is present of for the presence of alkaloid. Then Wegener test, reddish brown color with the precipitation of in the upper side. So this is show alkaloid. Then saponin test. Saponin is present only aqueous extract. Aqueous extract is reddish brown color, and its upper side foam formation of foam cause add of sodium carbonate. And then flavonoid test. So flavonoid is present only in methanol extract of seed with the precipitation of white precipitate. Then resin test only present in methanol extract. Then these pictures are gelatin lipid test. Lipid, there are three tests for lipid: gelatin, lead acetate, and ferric chloride. So in gelatin test, white precipitate is indicate the presence of lipid. Then violet or pink color in ethyl acetate, in the upper side and junction of two layers, this is indicate the presence of lipid. Then ferric chloride, light yellowish color in methanol extract, this is indicate the presence of lipid. And Salkowski test for steroid, reddish brown color in aqueous extract, this is indicate the presence of sterols. Then cardiac glucoside, there is only one test for cardiac glucoside, Killer-Kilani test in methanol extract, light yellowish brown color, this is indicate the presence of cardiac glycoside. Then primary metabolite screening in stem of Annona reticulata. So primary metabolizes carbohydrate, protein, and lipid. So there is a carbohydrate is absent in aqueous and ethyl acetate but present in methanol and petroleum ether, and protein is in stem part. Protein is absent all for extract aqueous, methanol, ethyl acetate, and petroleum ether. But lipid is present only in ethyl acetate and petroleum ether extract, but absent in aqueous and methanol both. There is pictures of these tests. So carbohydrates, there are three tests: Mollis, Benedict, and Fehling test. So reddish brown color, reddish brown color in methanolic extract show the presence of oligosaccharide, and in Benedict solution, light greenish color show the presence of polysaccharide. Then Fehling test, blue color indicate the presence of reducing sugar. Then protein test, so there are two tests: xanthoprotic test and biuret test. So protein is absent in xanthoprotic and biuret test. There are lipid test in methanolic extract with the white precipitate indicate the presence of lipid. Then secondary metabolite screening in stem of Annona reticulata. So secondary metabolite alkaloid है saponins, flavonoid, resin, tannin, gelatin, lead acetate, ferric chloride. These three tests for tannin, and sterol, Salkovsky test for sterol, cardiac glucoside, Killer-Kilani test, triterpenes, and anthraquinone. There are this is some pictures of this test. Then primary metabolite screening in leaf of Annona reticulata. So primary metabolites is carbohydrate, protein, and lipid. And then carbohydrate is absent in ethyl acetate and petroleum ether, but present in methanol extract of leaf. Protein is absent in xanthoprotic test and biuret test for extract of this plant: aqueous, methanol, ethyl acetate, and petroleum ether. Then lipid, there are three tests for lipid: solubility, glycerol, and sudon 3. So in aqueous and methanol, lipid is absent, but in ethyl acetate and petroleum ether, lipid is present. These are some pictures of these tests. Then secondary metabolite screening in leaves of Annona reticulata. Secondary metabolite, there are three tests for alkaloid, Mayer-Dragendorf, and Wegener test. So in ethyl acetate and petroleum ether, alkaloid is absent but present in only methanol and aqueous extract. Saponins, foam test for saponin, and saponin is present only in aqueous extract and absent in methanol, ethyl acetate, and petroleum ether. Then flavonoid is absent in aqueous, ethyl acetate, and petroleum ether, but only present in methanol. Then resins are absent in 4 extract. And tannin, gelatin test, lead acetate, and ferric chloride test. So tannin are absent in all 4 extract: aqueous, methanol, ethyl acetate, and petroleum ether. And sterol, so Salkovsky test for sterol is present only in methanol and ethyl acetate extract, but absent in aqueous and petroleum ether extract. Then cardiac glucoside, so there is a Killer-Kilani test for cardiac glucoside, and cardiac glucoside present only in petroleum ether and absent in 3 parts. Then Killer-Kilani test is only present in petroleum ether. Triterpenes only present in methanol extract. These are some pictures of this test. Then primary metabolite screening in fruits of Annona reticulata. Primary metabolites, carbohydrate, lipid, protein. Then aqueous, methanol, ethyl acetate, petroleum ether. So carbohydrate is present in aqueous extract and protein is absent all 4 extract, and lipid is present only in methanol and aqueous and ethyl acetate extract. These are some pictures of the test. Secondary metabolites screening in fruits of Annona reticulata. So secondary metabolite alkaloid. There are three tests: Mayer test, Wagon-Drop test, and Wagoner test for alkaloid. So alkaloid is present only in methanol extract. Then saponin. So saponin in present only in aqueous extract. Flavonoid only in present in methanol extract. Then resin are absent all four extract. Tannin, gelatin, lead acetate, and ferric chloride test for tannin. So tannin is present in only methanol extract. Sterol, Salkowski test for sterol. Sterol is present only in ethyl acetate extract and absent in aqueous methanol and petroleum ether extract. Then cardiac glucoside, Killer-Kilani test for cardiac glucoside. So there is cardiac glucoside present only in petroleum ether extract. Triterpenes only present in methanol extract, and anthraquinone are absent all four extracts. These are some pictures of this test. Total phenolic content of seed and stem. So value are presented as mean and standard deviation. And the total number of observation is 3. Then extract, seed, replicate. There is two part of this plant, seed and stem. So aqueous solution, first value is 791.13 and second value is 794.87 and third is 795.45. Then this is and its mean 793.82 and standard deviation is 2.34. Then for methanol extract, mean value is 53.98 and standard deviation 4 into 42. Then mean value of for ethyl acetate extract 255.38 and standard deviation 1.85. Then same it is stem part, leaf part, and stem and fruit part. This is graph of tannic acid concentration. Then total alkaloid and saponin contained in different plant parts, alkaloid and saponin, plant part, fruit, stem, seed, leaf. Fruit, stem, seed, leaf, and first value is and mean value is for fruit 1.577 and standard deviation 0.043. Then second value is 0.282 and standard deviation value is 0.028. Then mean value for seed 5.481 and standard deviation 0.068. Then mean value for leaf 4.667 and standard deviation 0.051. For saponin mean value is 0.164 and standard deviation 0.011 and mean value is for stem part 0.310, 314 and 0.037, 0.387. Then third is 0.016 and for leaf mean value is 0.882, 0.013. Same it is for flavonoid and terpenoids. Then this is graph show alkaloids, saponin, flavonoids and terpenoids percentage of these four secondary metabolites. So proximate analysis, moisture and ash percentage of different plant parts of Annona reticulata. So moisture, ash, carbohydrate and total fiber. Protein and fat percentage of different plant part of Annona reticulata. So protein, fat, sample, fruit, stem, seed, leaf, and mean value of fruit 9.23 and standard deviation 0.28, then mean value for stem 11.54 and standard deviation 0.24, then mean value of for seed 13.80 and 0.16 is standard deviation value, and mean value 14.26 and standard deviation 0.22. Same process in fat, same process is for fat content. And comparative profile of nutrition value of different parts of Annona reticulata. Moisture, there is fruit moisture is present 42%, then protein is present only 7%, then 4% is 4% present fat, and 5 and fiber is present only 5%, and carbohydrate is present only 33% in fruit, and stem stem in fiber is found in 70%, then protein is found only 11%, carbohydrate is found 3%, and ash is found 6%, fat is found in 5%, and moisture content found 5%. Then in leaf 29% fiber, 18% moisture, and 14% protein, then 30% fiber, then 12% ash, and 12% ash, and for seed 42% fat, and 30% carbohydrate, 13% protein, 9% moisture, and 3% and 42% fat. Then antimicrobial activity of different extract of seed and stem. So there is five bacteria. Three is gram negative and two is gram positive bacteria: Escherichia coli, Klebsiella pneumonia, Pseudomonas aeruginosa. These three are gram negative bacteria. Staphylococcus aureus, Streptococcus pneumonia is gram positive bacteria. So zone of inhibition only show in methanol and ethyl acetate extract, but not in aqueous and petroleum ether extract. Same it is stem. Zone of inhibition show in only in methanol and ethyl acetate extract, but absent in aqueous and petroleum ether extract. So same bacteria taken for leaf and fruit part. So there is a zone of inhibition present only in methanol extract for Klebsiella pneumonia, Pseudomonas aeruginosa, and Staphylococcus aureus, but absent in aqueous, ethyl acetate, and petroleum ether. Fruit part, zone of inhibition is present only in methanol extract, but absent in aqueous, ethyl acetate, and petroleum ether extract. There is a use of two control for antibacterial activity. Amikacin control is used for gram negative bacteria, and ampicillin control used for gram positive bacteria. And there are three bacteria of gram negative: Escherichia coli, Klebsiella pneumonia, and Pseudomonas aeruginosa. And gram positive bacteria is Staphylococcus, Streptococcus pneumonia. And these Petri plates of antibacterial activity with 4 extract, and with the amikacin and ampicillin control with zone of inhibition. These pictures are of all 4 parts: stem, leaf, seed, and fruit. Then antifungal activity, Candida albicans. Then antifungal activity, I have used Candida albicans Candida albicans fungus. Then miconazole control use as antifungal agent. There are four parts: seed, stem, leaf, and fruits. So zone of inhibition only show in methanol extract for seed and leaf, not stem and fruit part, and absent in aqueous, ethyl acetate, and petroleum ether. But miconazole acted on but methanol acted for only seed part. And these pictures of antifungal activity and interpretation of FTIR spectra for the associated functional group. There are some peaks. So some peaks, this is a smaller peak and carboxylic group, and 871.77. This is carboxylic group. And then 923.28 and 992.63. These both are aromatic ring, and 147 and 0.27. This is amide group, shown in amide group, and 1447.33. 33. This is nitro group, and 1515.16. This is alkanes group, and 100 1651.92. This is aromatic ring, and 1695.26. This is so for alkanes, and 2853.01. This is so carboxylic group, and 2926.63. This is so for carboxylic group, and ether group. This is so for ether group, and 3374.48. This is so amide group. And there are some peaks of functional group from the process of FTIR, Fourier transform infrared spectroscopy. Then discussion, the preliminary qualitative phytochemical screening of the crude powder of different parts of Annona reticulata plant was done to assess the presence of bioactive components. During the proximate analysis, high carbohydrate content was found in stem of Annona reticulata and fruit, which is high in sugar content. Seed of Annona reticulata have been a rich source of oil. Quantitative estimation of major phytochemical showed good presence of alkaloid, flavonoids, saponins, terpenoids in different plant parts. Based on the phytochemical analysis, nutritive value, and antimicrobial activity, select methanolic extract was fraction using gel filtration method and FTIR. Conclusion, in preliminary qualitative phytochemical screening among primary metabolites, presence of carbohydrate and lipid was observed in all four extracts of the plant, while protein was found only in methanolic extract of seed. Among secondary metabolites, extracts of different parts of plant contained flavonoids, saponin, alkaloid, and tannin, which is responsible for antimicrobial activity. The proximate analysis show higher percentage of protein, carbohydrates, and fiber, and low percentage of moisture and ash, make it a suitable food and food supplement. Characterization of partially purified biological active compound by Fourier transform infrared spectroscopy revealed the possibility of alkaloid compounds in the active fractions of the seed. Then thank you so much for hearing me. If any mistake, so please excuse me, forgive to me.